Hi, my name is Chris Katansky. I'm a postdoc in the Pan Lab, and today I'm going to tell you about some of our work with pseudouridine and in the immune regulation. So to start, I'm going to talk to you about pseudouridine biology. Then I'm going to review nanopore sequencing technology, and then finally dive into some of our results related to interferon treatment. So in 2020, I became a lot more educated about viral biology and host innate immune signaling, and part of that ended up being RNA modifications. So there's a number of cases where RNA modifications affect how cells sense and respond to foreign RNAs. Methylations and inosine modifications, there's a number of different stories. And so this biology is closely related to foundational work by Catlin Carrico. And so this is showing that synthetic RNAs decorated with different modifications help to dampen the immune response to transfected RNAs. So this is foundational to some of the mRNA vaccines that we benefit from today. So this got us thinking, do endogenous mRNAs also incorporate pseudouridine and these other modifications to evade immune recognition? So what is pseudouridine and why are we so interested in it? So pseudo-U is a modified uridine base. So the nucleoside is popped off, flipped around, and reattached via carbon-carbon bond. You'll notice that the Watson-Crick face is the same. So it's very challenging to detect pseudo-U by base pairing or sequencing methods. Pseudouridine is abundant. It's about as abundant as M6A in mRNAs. And like I mentioned, pseudouridine is very challenging to detect. So in 2014, 2015, 3 different groups published transcriptome-wide atlases using clever chemistry to detect pseudouridine by sequencing. And one of the striking results is the low overlap between all of these methods. If anything, this is a comment on the diversity and challenge of measuring pseudouridine biology. So we're looking for additional methods to help measure pseudouridine, and we turn to Nanopore. So if you're unfamiliar, Nanopore is a sequencing technique that uses a protein pore in a membrane. Nucleic acid is thread through the pore, and a voltage is applied across the membrane. So this drives ions through the pore, and the flow of ions is restricted by the nucleic acid. And different bases restrict the flow to a different degree. And we can measure this as a change in current over time. Now, Nanopore is really well suited to detect very subtle changes from bases like pseudouridine because of the richness of the data. So other groups have shown that with direct nanopore sequencing, you can detect M6A. However, pseudo-U can also be detected, but the features are much more subtle. So it's not just mutation and insertion deletion, but we're also looking at dwell time and base quality and other features of these traces that allow us to detect pseudo-U. Because the data are so rich from the nanopore, it screens machine learning problem. The trouble is, to predict pseudo-U using machine learning, we need a diverse training set. That way we can train the machine algorithm with as much data as possible. So to get this diversity, we turned to the human microbiome. So our setup uses rRNA, where we know confirmed pseudo-U sites, and we used samples from human yeast, C. elegans Drosophila, and human stool. And we combine the RNAs and split them for either Illumina style sequencing or nanopore sequencing. And so the Illumina sequencing helps us validate where the pseudo-U sites are. And the nanopore data is what we're going to use to train and predict our pseudo-U sites. Because we used such a diverse set of different RNAs, we find that we can cover 236 of the 256 possible 5-mers. So that's a pseudo-U with 2 ends on either side. And this diversity lets us train our machine algorithm with a broad set of data. The actual data looks like this. Looking at our Illumina sequencing, we use bisulfite chemistry to detect pseudo-U. So this produces a deletion rate. So with no treatment, we don't see pseudo-U, but with the bisulfite treatment, we get these abasic deletion sites. When we do nanopore sequencing of the same material, we find that we do get deletion sites at some pseudo-U positions, while mutation lets us see pseudo-U at other positions. We can get more granular and say mutation to C or mutation to A. Altogether, we used 12 different features, like these 3, to train our machine learning algorithm. So here they are ranked in order of correlation coefficient. So we've got insertion, deletion, misincorporation, base quality. And we found that the more of these features we use, the better our predictions get. So the blue trace shows just using one of these features. And as we incorporate more and more and more features into our algorithm, we find that the base calling becomes more and more accurate. So we're happy with the machine learning algorithm. We think that we can call pseudo-U. So what about the interferon-related stuff? So experimental setup looks like this. We have cells, and we either left them untreated, or we treated them with interferon gamma or beta. So these are central classic immune signaling molecules that elicit an antiviral anti-foreign RNA response in cells. We let the cells sit for 24 hours, and then we collected RNA and sequenced it through the Nanopore. And one of the most striking results is the correlation between induction of genes, just in abundance, and the incorporation of pseudo-U. So those data look like this. Here, each dot represents a single gene, and on the x-axis, we have fold change in abundance during interferon treatment. And on the y-axis, we have change in pseudo-U probability. So from our algorithm, every single U gets a score, some probability of being U or pseudo-U. And what we've done is we've taken the average of every U in that gene, and then we're looking at how that average score changes with interferon treatment. And the striking result is that the induced genes are the ones that have the biggest increase in pseudo-U. So it seems like the new synthesis, the induced genes are the ones that are getting pseudo-uridynylated during interferon signaling. We can flip the question around and talk about sites where the pseudo-U level has increased. So we can take the top 50 genes with an increase in pseudo-U level and ask what Go terms are associated with those genes. And we reproduce our result. We get interferon beta, we get response to viral infection, viral defense. So we get the same result. Now we worked really hard to replicate these results via a different method. So we developed a qPCR CMC-based validation here. So we're looking at a control gene, actin, and we're asking, when we treat it with cells with interferon, do we see any change in pseudo-U levels by a qPCR technique? And it's mostly flat. If we look at an interferon-stimulated gene, ISG15, indeed, we replicate that there's an increase in pseudo-U levels across the entire gene by qPCR. So what we think is going on is that the genes that are driven by an interferon signaling pathway, these genes are needed to drive an antiviral, anti-foreign RNA response, but the transcripts themselves must evade that same response. So what we hypothesize is that these genes are specifically getting pseudo-uridynylated so that they can avoid their own protein product degradation and signaling. Okay, so everything I told you was based on pseudo-U calling at individual sites, based on many reads that map to an individual site. However, Nanopore is fundamentally a single molecule technique. So instead of assigning a score to any particular site, we can actually assign a score to every individual read, every molecule that maps to that site. And so that lets us ask different questions. In particular, we can ask how pseudo-U sites on the same molecule talk to each other. So the simplest case is comparing independent and dependent sites. So this is an easy example where there's an upstream and downstream site. They're both 50% modified. And if you subset just the ones that are modified at the first position, you still get a 50% at the second position. So they're independent. The obviously different case is dependent sites where they're both 50% modified, but if you're modified at position 1, there's a 0% chance of being modified at position 2. So what do the real data look like? Here we're looking at a heat map. Every row represents an individual read from our Nanopore sequencing, and the color is the Nanopore probability given from our machine learning algorithm. And we can see sort of all 4 categories of reads. We see positions where both sites are modified, one site is modified but not the other, vice versa, and neither site is modified. And these happen in about the right probabilities, so we can tell that these are independent sites. The contrast is dependent sites. So there's sort of obviously something missing. We've got one site modified but not the other, neither site modified, and one site modified but not the other, but we never see both sites modified when looking at these 2 positions. So we know that these are dependent sites. And we can get a p-value by looking at KS test between the probabilities here. And so I can do this calculation for every pair of sites in a transcript and ask how they are connected to each other. And sort of your intuition is that they're not really going to be connected. They're independent sites. Many of them are very distal. And that's actually the result that we get. We see a lot of gray squares indicating that the p-values are not significant. Everything along the diagonal is perfectly correlated with itself. And we get a few large boxes, which are proximal sites that are correlated with each other. And that's the example I just showed you here. What is absolutely wild is once we do the interferon treatment, we see expansion of pseudouridine zones that are all correlated, but then the appearance of these very distal sites that are highly correlated with one another. So these sites are separated by thousands of bases, and yet modification at one position is highly correlated with modification at the other at the single molecule level. So we're really excited by this sort of perspective. We think it's a new way to think about these modifications, especially the biology that they're doing. So we're excited to have this new analysis platform to let us ask these sort of questions. So thank you for listening. Conclusions for today are we developed a Nanopore analysis pipeline called NanoPsu that lets us detect pseudo-U directly from Nanopore RNA sequencing. We found that pseudo-U is incorporated into interferon-induced transcripts. NanoPsu also lets us detect pseudo-U at the single molecule level. And using this, we found that pseudo-U sites can be linked across kilobases, indicating some coordination of biology that we don't yet fully understand. So acknowledgements. I had the honor of presenting this work today, but it was very much spearheaded by a graduate student in the lab named Sihao. Microbiome work was done with Karen from the IRIN lab, and the interferon treatments were done by Devin from the UDEL lab. Thank you for your time, and happy to answer questions via whatever medium makes the most sense. Thank you.